Thank you very much. I want to join the previous speakers and thank you all for the invitation. It feels as a privilege to be here, and not only are the talks very stimulating, also you're treating us very well in this beautiful city. So thank you again for that. So as many of the previous speakers, I work in the field of developmental or fetal programming. I'm not going to talk too much about this concept because it has been introduced so well. I just want to highlight one thing. So among the many conditions that have been identified that impact, or that have been identified during pregnancy that impact health and disease risk in later life, it's important to note that these conditions do not per se cause disease, but they change the susceptibility or vulnerability to later diseases. So we all know that the probability to acquire a complex common disorder is a function of exposure to risk factors, like, for example, traumatic experiences or a bad diet or a lack of physical activity or smoking, and our susceptibility, because not all people, as we heard yesterday, that are exposed to a trauma actually develop PTSD. Not everyone who is smoking and eating bad food is developing a heart condition. And this susceptibility in traditional medical models is always equated with the genetic makeup of a person. But there's now more and more evidence that processes very early in life, in conjunction with our genetic makeup, can change structure and function of physiological system that then can shape our susceptibility. And given the environment we are exposed to later in life, we might acquire a disease or not. So this is one of the core promises of the fetal programming hypothesis, and it's important to highlight it because this is where telomeres come into place later. So we work in the area of maternal stress during pregnancy. How does it impact fetal development and disease susceptibility? And it's now fascinating to think about how maternal stress can actually impact fetal development physiologically, because maternal stress is something that originates in the maternal brain. And there are no direct neural connections between the maternal brain and the fetus. So every communication, biologically speaking, from the mother to the fetus has to go through the placenta and the cord blood. And the placenta, and not many people, of course, here, they know it, but in other audiences, they don't know it. The placenta is actually an organ of fetal origin, and we heard the placenta has a sex, and it's the sex of the fetus that the woman is carrying. And the fetus uses the placenta as an information system to acquire information about the intrauterine and extrauterine environment. And the fetus also uses the placenta as an effector organ. It can release biological signals to the maternal compartment, and we even have data to suggest that the fetus, as it or he or she develops, can actually alter the maternal stress response during pregnancy. And it's now interesting that the communication pathways, or the biological mediators that are used between the mother and the fetus to communicate, are in part the same biological agents that are used to regulate the stress response, the biological stress response. And this is a very oversimplified model how we explain maternal effects on fetal, or maternal stress effects on fetal development. As we all heard already, if the mother is stressed, she releases cortisol, and this cortisol can in part can pass through the placenta. There's an enzyme that converts part of it into the inactive form, cortisone, but about 20%, it's estimated, can pass through the placenta into the fetal compartment and then have programming actions, for example, on the fetal brain, on fat tissues, and so on. We also know that under chronic stress conditions, there's more inflammation in our system, and the pro-inflammatory cytokines that are increased under chronic stress conditions, also during pregnancy, they might not cross the placenta because they are too large, the molecules are too large, but they might induce inflammatory processes in the placenta that then increase inflammation in the fetus. And also in acute stress situations, we release epinephrine and norepinephrine, which leads to vasoconstriction of the placenta. So the fetus gets less of nutrient supply, which again is a stressor for the fetus. And together, all of these processes can increase biological stress mediators in the fetal compartment that then can program different fetal physiological systems as they are developing. So in the very first study that we conducted many years ago, we wanted to see whether maternal psychological stress actually is associated with increased disease susceptibility for common complex disorders in later life. And this was really the first study to look at this, and we used a retrospective case control design, and we operationalized stress during pregnancy as major stressful life event that happened to the mother while she was pregnant. For example, loss of a mother-child, loss of a partner, severe disease of the partner, and so on. And we recruited a comparison group whose mothers did not report any such events during pregnancy. And we studied young adults. So these adults were not sick yet. So we focused on pre-disease markers of physiological dysregulation that are altered before the actual disease occurs. So, for example, we looked at metabolic function. We did an oral glucose tolerance test with the participants. We looked at the immune system after PHA stimulation. The participants underwent a psychosocial stress test in the lab because we wanted to stimulate their HPA axis, and they did a cognitive working memory test. And it was kind of shocking to see that we found alterations in all of the different physiological systems we looked at based on maternal stress exposure during pregnancy. So these young adults showed increased body fat, they showed higher BMI, they had higher insulin levels 2 hours after the oral glucose stimulation, suggesting insulin resistance. They showed an altered endocrine profile after psychosocial stress exposure, and we also saw an immune profile that makes them more prone for allergies and atopic diseases, and they had impairments in their working memory, performance, or executive function. So taken together, we saw alterations in physiological systems that predispose for a range of aging-related diseases later in life. And this was a retrospective study, and methodologically it's not ideal to assess stress 25 years after it has happened, because many things can happen in between. So that's why ever since I did this study, I got involved in prospective birth cohort studies, in which we usually recruit the women relatively early during pregnancy. We try to do 2 to 3 assessments during pregnancy, in which we characterize psychological stress and other experiences of the mother with questionnaires and interviews. And we also collect different biological samples to analyze biological stress mediators. And in our more recent studies, we also established a protocol to assess stress in an ambulatory manner in everyday life. So the women carry smartphones on which we prompt them several times during the day to ask them, how stressed are you feeling right now? How well supported do you feel? What have you eaten and drunk since the last prompt? And whom are you with right now? They also collect heart rate monitors so that we can look at heart rate and physical activity, and they collect saliva samples so that we can look at cortisol production in their everyday life. Can I go back one slide? Yeah, there. And then at birth, we try to collect placental tissue and cord blood, and then we follow up the children. Right now, in the most recent Berlin study, we have funding to follow up the children until the age of 4 years. And we're interested mainly in 2 outcomes in the children. One of them is brain development and cognitive function, and the other one is obesity risk and risk for metabolic disorders. And since cortisol is so important for the transfer of the stress effects on the fetus, we developed a quite involved protocol for the mother to collect cortisol samples during pregnancy. Cortisol has a diurnal shape. It increases by 30 to 50% after awakening, and then it decreases over the day. So if you want to have a reliable, if you want to make a reliable statement about cortisol, you really have to try to capture different aspects of the dynamic of the diurnal variation of cortisol. So the women collect the first sample immediately after awakening, the second one 30 minutes after awakening, and then they collect 3 samples across the day. And we do that at 4 days in each pregnancy trimester, so we get an average of 60 samples per woman across pregnancy. And these are just some of the findings that we have with maternal cortisol during pregnancy. So we could show, and this is mainly work that my colleague Claudia Buss has been doing, that more cortisol production during pregnancy, higher levels of cortisol during pregnancy are associated with a higher amygdala of the offspring. And this was measured with MRI scan at 7 years of age. And we see the same already in newborns that have not been exposed to any postnatal environment or stressors. So we see a bigger amygdala already at the newborn age, and the amygdala is connected differently to other areas of the brain when the mother has been exposed to high, or when the mother released a lot of cortisol during pregnancy. And the amygdala is critical for emotional processing, and the size of the amygdala has been associated with risk for anxiety disorders later in life. The other outcome we're interested is obesity risk. So in one of our studies, the children underwent DEXA scans at 0 and 6 months of age, and we could look at fat deposition during this very early critical period of development. And what we saw is that the more cortisol the mother produced during pregnancy, the higher the fat deposition of the baby during the first 6 months of life, and this might be a phenotype that is advantageous in an environment where there's not enough nutrition, but in our westernized societies, this might make them more prone to obesity and metabolic disorders later in life. So the developmental programming field has brought forward a host of different conditions during pregnancy that are associated with several different health and disease-related outcomes, including alterations in the metabolic, endocrine, immune, cognitive, and affective system, and so on. And how are these effects actually biologically embedded? This is still an area of intense interest, and research is done in this area. So there are possibly two possible mechanisms here. One of them is there are outcome-specific mechanisms. So for each of these different outcomes that are affected, you have one particular mechanism. And in addition, there might be something more common underlying physiological mechanisms that might drive changes in all these different physiological systems. And of course, epigenetics has been discussed, rightfully so, as a big player in this. And we put forward the hypothesis that alterations in telomere biology might, in addition to all of these other processes, represent one of these mechanisms underlying risk for so many different diseases based on prenatal exposures. So what are telomeres? Most of you will know this, but just very shortly, telomeres are non-coding DNA sequences at the end of our chromosomes. They're critical for chromosomal stability during certain phases of the cell cycle. And the interesting thing about telomeres now is that the DNA polymerase machinery is not able to fully replicate the DNA strand after cell division, and that's why telomeres lose base pairs with each cell replication. And eventually, telomeres reach a critical short point at which the cell can no longer divide, and the cell then either dies or enters a state of senescence. And senescence cells show all kinds of morphological changes, including changes in their chromatin structure and chromosomal fusion and so on. And senescent cells also show an altered gene expression profile, more prone to a pro-inflammatory gene expression profile, and thereby they can alter neighboring cells and tissues, which leads to this perpetuatory cycle of the aging process. And besides regulating or putting a limit to the replicative capacity of the cells, telomeres are also very important for the integrity of the overall chromatin structure, and they can actually function as a epigenetic mechanism, because the telomere end can loop back and touch the chromosome, and while the telomeres shorten, more of the chromosome more of the DNA becomes available for gene expression. And this is how shortened telomeres actually can work as an epigenetic process to alter gene expression in the cell. Telomerase is a very important enzyme in this context because it can actually counteract telomere shortening. It can add back the DNA pieces after cell replication. It stimulates cell proliferation. It's very important in very early development because it's involved in many signaling pathways during tissue synthesis. Telomerase can travel outside the nucleus, get into the mitochondria, and reduce oxidative stress there. So there's a bidirectional interaction between telomeres and the mitochondria, and it also improves energy efficiency of the mitochondria. So overall, it's a very important enzyme that promotes cell resilience. Because telomeres are so important for cellular function, it's not surprising that shortened telomeres in human studies, mostly measured in immune cells, are associated with longevity and a host of different diseases. And there are more than 50 meta-analyses published right now that show links between telomere length and a range of different physiological and mental diseases and health behaviors, including, for example, cardiovascular diseases, obesity, Alzheimer's, depression, smoking, physical activity, and diet. There's an ongoing debate whether telomeres are a biomarker of the aging process or whether they are actually causally related to pathology. Evidence that they are causally related comes from monogenetic disorders where you have mutations in genes that are important for telomerase expression, for example. And also, very interestingly, in organ transplantation studies, it could be shown that survival time after organ transplantation and the disease progression are related to the telomere length of the donor and not of the recipient, which also is one more hint that they actually are causally related to certain kinds of pathology. There's more and more evidence and recognition in the field that the way we start with in terms of our telomeres matters for later aging. So the length of the telomeres at birth are critical for later telomere length and telomere attrition rates. We know that there's large inter-individual variation in telomere length at birth. It's actually larger at birth than at later stages in life. And the decline in telomere length in early life is more rapid than in later life. Speaking of sex differences, telomeres are consistently been found to be longer in females than in males, and we already see these differences, these sex differences at birth. And the initial telomere length at birth is related to telomere length later in life. And in a bird study where they could track telomere length from birth to death and related to longevity, they could show that newborn telomere length is a far better predictor of lifespan than telomere length at later stages of life. And it seems that individuals maintain their relative telomere rank across the lifespan over several decades, which again highlights the importance of the initial or newborn length of the telomeres. Despite the importance or increasing recognition of the importance of the initial telomere length, the determinants of newborn telomere length are still very poorly understood. Of course, genetics plays a role here. Heritability estimates for telomere length range between 0.6 and 0.8. However, known genetic variants only account for a relatively modest proportion of the variance in telomere length that we see, especially at birth. And there is interesting data suggesting that telomere length is more strongly related to maternal than paternal telomere length. And another study suggests that the currently identified loci that are associated with telomere length might not influence the initial telomere length, newborn telomere length so much, but may play a role in telomere length modification during the life course. And a twin study could show that the main determinants of telomere attrition over the life course were early life environmental factors and to a much larger degree than actually heritability. And all of this highlights the critical importance of environmental and maternal factors in the initial setup of the telomere system. And so why do we think this is important in the fetal programming hypothesis? So for any given individual at any given time point in life, telomere length represents the initial or newborn telomere length. It represents the telomere length attrition over time with age and the counteracting effects of telomerase activity. And if two newborns start with different telomere length, the newborn with the shorter telomeres reaches the critical point, the critical short point at which the cell system enters senescence at an earlier age than the individual that starts with longer telomeres at birth might render this individual more susceptible for aging-related disorders at an earlier time point in life. And so it's very important to identify factors that influence the initial setting of telomere length, and there's more and more evidence that conditions in prenatal life may actually program the offspring telomere system. And this is also reflected in the increasing number of publications on newborn telomere length since 2000. So what's the evidence that prenatal factors can program the telomeres of the offspring? What do we know about it? The very first evidence actually came from studies that Suzanne Ozan, who gave a presentation yesterday here, conducted in her animal models, where she could show that nutritional manipulations during pregnancy are associated with telomere length of the offspring. And there have been a range of different obstetric conditions during pregnancy, for example, preeclampsia, low birth weight, and so on, that have been associated with shorter telomeres of the offspring. Also, maternal BMI is associated with shorter telomeres. And there's one study from Japan, and I mentioned it because of the talk this morning, that showed that assisted reproductive technology, or pregnant in newborns of mothers that used ART to become pregnant, the telomeres at birth are actually shorter than in naturally conceived pregnancies. What's the evidence that maternal stress plays a role here? I'm finally coming to that now. So in adults, it's pretty well established that stress can shorten your telomeres, and there's a recent review paper about it that shows that several different kinds of stress exposure, like stressful life events, work-related stress, or financial strain, and different aspects of the stress response are associated with shorter telomeres. And also in children, it could be shown that severe stress exposure, like childhood abuse, other forms of early life adversity, are associated with shorter telomeres, and those changes actually drag into the adult life. And the mechanisms how stress induced telomere shortening are quite well understood, because if you incubate, for example, immune cells with glucocorticoids, with cortisol, it inhibits the activity of the enzyme Telomerase, and also other biological agents that are related to stress, like pro-inflammatory cytokines or oxidative stress processes, can damage the DNA in the telomeric regions and thus shorten telomeres over time. So I told you about this study where we recruited young adults with prenatal stress exposure and without. And we had the hypothesis that prenatal stress exposure might impact the offspring's telomere length. It hasn't been shown before. And we set up a collaboration with Elisa Apple and Elizabeth Blackburn at UCSF, and we sent them DNA that we had left over from the study, and they analyzed the telomeres for us, and we could indeed show that young adults whose mothers were exposed to severe stressful life events during pregnancy actually had shorter telomeres in their leukocytes, which might suggest more rapid cellular aging after being exposed to prenatal stress. And we replicated this finding in one of our prospective cohorts where we could already show an association between maternal stress exposure during pregnancy and telomere length in newborns in cord blood. And this association between various forms of maternal stress exposure during pregnancy and offspring telomeres has been replicated since then by various research groups around the world. And we are actually just about to submit a meta-analysis on the effects of maternal stress on offspring telomere length, together with Stacy Jury from Tulane University, whose student conducted this work. We identified 9 different studies that met the quality control threshold that we set for this meta-analysis that all showed an effect of maternal stress on offspring telomeres. So as I said, it's now interesting to study the biological processes that underlie the effects of stress on offspring disease-related outcomes. And it's known that chronic stress exposure is associated with pro-inflammatory cytokines. Pro-inflammatory cytokines, on the other hand, can affect telomere length and telomerase activity via NF-kappa B-mediated pathways. That's known from animal studies and from adults. And there are also mouse models that suggest that if you administer TNF-alpha during pregnancy, it's associated with shorter offspring telomeres in different tissues. So in one of our prospective pregnancy cohorts, we were able to quantify levels of pro- and anti-inflammatory cytokines during pregnancy, and we built a ratio of TNF-alpha over IL-10 to represent pro- and anti-inflammatory cytokines, and in a healthy system, it should be balanced. The higher the ratio, the more pro-inflammation you have in your system, and we could show that the higher this ratio, the more pro-inflammatory the milieu during pregnancy, the shorter the telomeres at birth. And let me just say a few words about cord blood. Cord blood, of course, is a mix of different tissues, and variation in telomere length has been shown in different blood cell types. However, in cord blood, there are very high correlations of telomere length and various hematopoietic cells, and also there's high correlation at birth in telomere length in different newborn tissues, for example, between cord blood, umbilical artery tissue, and foreskin tissue. So we can assume that the magnitude of variation in telomere length between individuals exceeds that of the variation in telomere length among cells within the individual, and that's why we think umbilical cord is a good tissue to look at effects of prenatal factors on telomere length of the offspring. So because it has been discussed a lot at this meeting, I want to present you as findings of a study that we did on folate concentrations and newborn telomere length, and that this also brings us a little bit to the bedside part of this meeting. So what can we actually do? Can we reverse? Can we prevent these severe effects that we see of these different exposures on offspring physiology? In one of the studies that was actually based at McGee at the University of Pittsburgh that we were part of, we were able to measure folate concentrations in pregnancy, and we measure telomere length at birth again in cord blood. And you all know that folate is essential for establishing the fetal DNA structure and epigenetic marks, both of which again are important for establishing the telomeres. And the developing fetus is completely dependent on maternal supply for its folate requirements. And in adult men and women, studies showed that folate concentrations are actually associated with telomere length. And we replicated this in our pregnancy study. So we found that the higher the maternal folate concentrations during pregnancy, the longer the telomeres at birth. And I'm coming to my last finding, which I put at the end because I all want to give us hope that there's something positive out there, positive message that we might be able to modify this and counteract the adverse effects of maternal stress during pregnancy. Because in one of the studies, we were able to look at maternal psychological resilience, which can be defined as the resilience or the resilience that we have when we are facing large amounts of stress. And it has also been defined as the healthy and positive function that we still have left over after we underwent a trauma or a stressful experience. And it has been argued in the psychological literature that positive emotions are more than just the absence of negative emotion. It's more than just the opposite of stress, because they play an essential role in creating resilience. And positive emotions, social support, psychological resilience, all have been associated with a more rapid recovery biologically from stressful experiences, and also with a smaller impact of stress on our biological system, which is represented by this allostatic wear and tear concept. So to look at the effects of maternal resilience, we were able to collaborate with a Finnish group, actually with the Pareto cohort that has already been presented to us. And this cohort is great because it's rather large. And in that cohort, I mean, the aim of the study was to look at factors that predict and prevent preeclampsia. And one of the hypotheses was that psychological stress might play a role here. So they assessed psychological stress and other psychological factors up to 16 times during pregnancy in this study. And we were able to analyze telomere length in more than 660 newborns of the study. And based on all the positive psychological constructs that were assessed in the study, like positive affectivity, positive mood reactivity, social support, we statistically built a positivity factor, and based on all the stress scales and the negative mood scales, we built a stress factor. And we then regressed the stress factor on the positivity factor statistically, and to build this resilience factor. And what it represents is the amount of psychological positivity that we have left after accounting for the effects of stress that we are facing. So this is, and then we entered this resilience factor in our regression model. And first of all, we replicated the effects of our previous study, now in a much larger cohort, that maternal stress is associated with shorter telomeres of the offspring. But we could also show that maternal resilience, the higher the resilience the mother reports during pregnancy, is associated actually with longer telomeres of the offspring, which again highlights the importance of maternal mental well-being during pregnancy, and that we care for pregnant women. And which leads me to the conclusion of my talk. There is, as you all know, evidence from several independent studies that maternal stress and stress hormones during pregnancy are associated with offspring disease-related outcomes. And I hope I could convince you that telomere biology might be a small part of the big puzzle that explains the developmental programming effects. The clinical significance of the effect sizes, I mean, differences in telomere length at birth, really needs to be established in future longitudinal studies that we are now working on. We also need replications of some of the aspects that we observed in other cohorts, and we need to use more refined methods to assess telomere length. There's a big debate in the field about that as well. However, I think these findings are critical because whatever we observe during pregnancy has an impact not only on the mother, but also on the fetus, the next generation, and maybe even more generations to come, because although transgenerational transmission hasn't been proven in humans yet, there is no reason to believe that it shouldn't work in humans. And when I see all the refugees from the Ukraine coming into the Berlin train station, several thousand every day, most of them are women, a lot of them are pregnant, this again highlights the importance of this whole topic, and that we do something for mental health of pregnant women to improve health of future generations to come. And with that, I want to thank my collaborators. The studies we are conducting are very labor intensive, so it needs big teams and groups, and it needs international collaborations to study different aspects of what I have been presenting here. My thank goes also to the funding agencies that have been continuously supporting this work, and I thank you for your attention.